All right, thank God.
Does that need to be dragged over? Not the video. All right, folks, we will go ahead and get started. I'm really excited to introduce today Kaylee Arnold. Kaylee is originally from San Diego and earned her bachelor's degree in biology at University of Redlands. She later completed her master's degree in EEB at Tulane University and then began her PhD at the University of Georgia in the Odom School of Ecology. During her early graduate studies, Kaylee became interested in exploring the impacts of anthropogenic disturbances on host-associated microbial communities and infectious disease transmission. As a grad student, Kaylee also regularly volunteered with public K-12 schools in Athens, Georgia, and co-led the organization EcoReach, which brings students and faculty into classrooms to teach various ecological topics about careers in ecology. Kaylee just defended her dissertation a few months ago and is the newest Living Earth Collaborative postdoc. Today, she's excited to discuss her dissertation research and her upcoming plans at WashU. So please join me in welcoming Kaylee.
All right. Thank you for coming, everyone. I'm very excited to share with y'all the work that I did for my dissertation. So as Kara said, I went to the University of Georgia in the Odom School of Ecology, and I worked with Nicole Gottjanger. And yes, I just defended in November, so it's all still a little bit of a blur. And then so today, I will discuss disease vectors and the role of their symbiotic organisms. I will discuss my study system, Chagas disease. And then I'll go into some detail about like 2/3 of my dissertation research. And then very briefly, hopefully I won't run out of time, but very briefly, I will talk about some of my proposed plans for this new postdoc. So the microbiome, I'm sure you all heard a lot about it. I know we just had a seminar speaker yesterday talking a lot about the gut microbiome. So one thing I like to kind of like initially start out with is just saying microbiome versus microbiota. Sometimes I use them interchangeably, but they are different. So the microbiome consists of all the genetic material of the organisms you're looking at, versus microbiota is really just looking at kind of presence, absence of the microbes. So for my dissertation work, I really just look at the microbiota. So it's really just presence, absence of bacteria is what I kind of focus on. So why do we care about the microbiome? Well, so at least for the gut microbiome is responsible for a lot of different important processes. So that can include digestion, nutrient uptake, stress tolerance, reproduction, behavior, and pathogen resistance, which is why I'm particularly interested in it. So I study disease vectors and transmission risk. And so the main big driving question of my dissertation is what can influence a vector's ability to transmit infectious diseases? So first, what is a disease vector? So a disease vector is any organism that can spread disease. So that will include your mosquitoes, ticks, kissing bugs, fleas, and they can transmit pathogens that cause malaria, Chagas disease, guinea worm Lyme disease, and they are responsible for up to 17% of all infectious diseases. So again, I think about what shapes vector-borne diseases. So I actually look at all 4 of these factors. So I really think about the pathogen itself. I think about the vector, the environment. I am an ecologist, so I do think about the environment, and then finally the host, so what the vectors are feeding on. So now, what role do microorganisms play in vectors specifically? So I'll just kind of give 2 examples real fast. So development. So, so some papers in the 40s and 50s found that if you clear the gut symbionts from triatamines, then they do not develop into the adult stage. And then also, researchers found that mosquitoes with greater gut microbial diversity were less likely to be successfully infected with malarial parasite in a lab experiment. So now let's go to my study system. So Chagas disease. So Chagas disease is found throughout the Americas. 8 to 15 million people are infected. It is vectored by kissing bugs or triatomines, and is caused by a protozoan parasite, Trypanosoma cruzi. So I'll just kind of get really like basic into the transmission cycle. So first you'll have a triatomine or a kissing bug. And then one thing to note is that triatomines are obligate blood feeders, and they can feed from all vertebrates, but only mammals are competent hosts for Trypanosoma cruzi, the parasite. So first you have an unaffected kissing bug. And then here you have a mammal. So this is an infected mammal. I'm infected with T. cruzi. And then now you'll have a kissing bug will feed from this infected mammal. And the kissing bug will pick up the parasites and become infected and infectious. And then this infected triatomine will feed from a new mammal. And then triatomines will defecate infected feces on the mammal. So this is kind of a big part of the transmission. So you only have a successful kind of transmission event if the infected feces gets into the bloodstream of the mammal. And then so those infected feces went to the bloodstream and then cause the animal to become infected. And then that completes the cycle. And something, this is kind of an aside, but so the time between like for the triatomine feeding from the mammal and then also defecating, that plays into transmission. If it takes them too long to defecate, then the animal might get away, and then you won't have the continued cycle. So it is important to kind of think about how quickly they do defecate and then how likely the feces can get into the animal system. So triatomine. So I was really interested in looking at their gut microbial communities. So as I said, they are obligate blood feeders, which means they have a nutrient-poor diet. So they require specific gut microbes to synthesize essential missing nutrients. And then gut microbial diversity may be associated with decreased parasite infection. So if you can kind of think back to those two examples about gut symbionts and disease vectors, I'm kind of thinking about so they require missing essential nutrients and microbes to synthesize them. So it's probably likely why when you clear the gut of microbes that the triatomines don't develop into the adult stage, which has an impact on transmission and reproduction, just a continued cycle of Chagas disease. And then also kind of thinking about gut microbial diversity and what that means for parasite infection or parasite resistance. And then finally, a lot of gut microbes are acquired from the environment. So something that I'm really interested in is looking at disturbance and how does disturbance like deforestation have an impact on vector competence. And vector competence is a vector's ability to acquire, maintain, and transmit a pathogen. So my overall objective is to understand the associations between Chagas disease vectors, their associated gut microbial communities, trypanosome infection, and land use change. And then so my first question, is a region or habitat type associated with gut microbial diversity? And then also, is trypanosome infection associated with gut microbial diversity? So for the bulk of my dissertation, I worked with wild triatomine populations. So we worked in Panama. So Rhamnius pallescens is the primary vector in Panama. Rhamnius pallescens are found in the crowns of royal palm trees. So this is why it's really important to think about the environment and any kind of disturbance happening at the landscape level. There is a quickly changing landscape, so you have a lot of land use change happening throughout central Panama. And then studies have found that there is an association between deforestation and then you have increased R. pallescens abundance, as well as increased T. cruzi infection rates with deforestation. So I'm trying to like stand back a little bit and think about sort of my whole project and why I care about both kind of disturbed landscapes and gut microbial communities and infection of the parasite. So you'll have so you have undisturbed landscape and now you have a disturbed landscape. So we can kind of think about deforestation here. And then so disturbed landscapes may alter host composition. So you might have difference in host richness, maybe availability of hosts in these disturbed landscapes, which will have an or may likely have an impact on tridamine abundance and infection rates. So their ability to find a blood meal, how quick is that? There is some evidence that different like smaller mammals may have like increased kind of the parasite can replicate quicker in some different types of mammals. So thinking about what they're feeding on and again how frequently they're feeding on them. And then thinking about particularly infection. So there is a lot of research showing that there are some impacts of infection and gut bacteria. So the gut bacteria does interact with the parasite and the tridamine bugs. So if you have different infection rates, how does that have an impact, especially at the population level on the tridamine gut microbial communities? And then further, also, how does that then have an impact on infection if their microbial communities are altered? Perhaps that may have an impact on the ability to pick up a subsequent infection. And then also we can look at, so we have our altered host composition again, and also that might just have an impact on the gut microbial communities. There's a lot of research looking at diet, so in this case, blood meal diet and their gut microbial communities. So what does that mean if we have shifting host composition for their microbial communities? And then again, infection. So if you have shifted gut microbial communities, maybe differences in gut diversity, what does that mean for infection? And then finally, we can also think about some environmental microbes. So if you have an altered landscape, perhaps you're going to have altered environmental microbes. And then ultimately, what does that mean for the gut communities as they do pick up a lot of their gut microbiome from the environment? And then again, finally, what does this mean for transmission? So back to my kind of first main question. So is region or habitat associated with gut microbial diversity? And then so I want to investigate the association between landscape variability and gut microbial composition and diversity. So, so back when I was a baby grad student, so we sampled throughout central Panama. So we sampled across 3 different types of palms. This sort of represents our deforestation gradient. So we sampled across pasture palms, paradomestic palms, and forest remnant palms. So our pasture palms are our most disturbed, and our forest remnants are going to be our least disturbed. So we sampled in 2 kind of main areas at 3 sampling sites. So first we collected samples at 2 main sampling sites right around the Panama Canal, and there we collected pasture palms and paradomestic palms. And then finally we sampled, it was about like, I think about an 8-hour drive from the Panama Canal. It had a higher elevation, more precipitation, just a very different landscape. And then there we collected samples at pasture palms, paradomestic palms, and then also at forest remnant palms. So, so just some of my field work. So we went and so we would put traps into palm trees. So basically our neuro trap is like the T end of a PVC pipe with double stick tape on it. And then we put a live mouse inside. We would kind of hoist it into the crowns of the palm trees, wait about 3 days. And then hopefully some of the kissing bugs would, on their way to try to get the mouse, would get stuck on the tape, and then we would be able to collect them. So in total, we collected 288 R. pallescens across central Panama. Each sample had its mid and hind guts dissected. And then we extracted the DNA from the tissues and then sent everything out for 16S sequencing to look for the bacteria as well as 12S to identify the blood meal host species. So just kind of starting to look at our results. So if you remember, we had 2 samples around the Panama Canal, 2 sampling sites around the Panama Canal, and then 1 site a little bit further out called Veraguas. So this is looking at the mean relative abundance of a bacteria at the genus species, at the genus level. So the biggest main takeaway is out of 100%, like what percentage of each different bacterial taxa we're seeing. And really the biggest takeaway is that the 2 canal sites look quite similar, just visually, compared to the Veraguas sites. And then so here we're looking at alpha diversity. So that is within sample diversity. And again, we're seeing differences between Veraguas and our 2 canal sites. So here our Veragua samples have greater alpha diversity compared to samples at the canal sites. And then here, so this is beta diversity. So here we're looking at, this is between sample. So each dot represents a different individual bug. And so again, the canal sites have a similar community structure compared to Veragua's. So you can see a lot of overlapping, and that means that the overall bacterial community is quite similar. And the biggest overlaps we're seeing is between Capira and La Trera, which is our two canal sites. That's that big clump right there versus our Veragua sites, much less or more dissimilar and a bit different from our other sites. So again, we're saying that the canal sites are quite different. And then so now we also want to look at habitat type and the type of palm. So here we're comparing the pasture, paradomestic, and forest palms. And we're seeing that our pasture and paradomestic samples have a similar bacterial composition. So again, we're seeing a lot of similarities with these pasture and paradomestic palms and quite dissimilar overall composition looking at our forest samples. And then again, looking at our alpha diversity, so we have our forest samples that have greater diversity compared to the pasture and paradomestic palms. And then there's no difference between our pasture and paradomestic palms. So now just kind of looking at only Veraguas. So we know there's differences between Veraguas and the two canal sites, but now let's try to hone in a little bit more on the habitat type. And so here, only at Veraguas, we are still seeing differences between our forested sites and our pasture and paradomestic sites, and then no differences between the pasture and paradomestic. And then looking at this beta diversity again, we are seeing a lot of clumping between the pasture and paradomestic, that's your green and blue coloring, and then quite a bit difference with our forest sites. So we're seeing differences between like where we're sampling, and then also differences in the type of habitat we're looking at. So what are driving these differences? So the main thing that at least I'm able to look at is looking at the host blood meal. So for this figure, so it says number of blood meals. Really think about that as just kind of individual samples. So each tritamine, we looked at the dominant blood meal they fed on, so the one that had the most sequencing reads attached to it. So here we can see that if you're just looking at kind of like percentages, you can see that the Capira and La Trera, our two canal sites, do look quite a bit similar. We're seeing like a bit of red, a lot of that lighter red color, and ultimately quite different from our samples at Veraguas. So that's just looking at the different blood meal host species that they're feeding on. And then same thing for our forest versus our paradomestic and pasture palms. You're seeing quite a bit difference between the three of them. And then I'm thinking quite at the forest side, you're really only seeing these two species. And then again, looking at this relative abundance. So I think this is maybe a little bit more clear, but so samples that fed on Didelphis, which is large American opossum, looks quite a bit different from the other two. And then Heteromenes is a small rodent, and then we have our philander, which is another opossum species. So we're seeing some differences just based off, this is just the dominant blood meal. So perhaps this is driving at least some of the differences we are seeing across both sampling area as well as habitat type. And then finally, just looking at alpha diversity. So I have all the diversity metrics on here, but you can call it as a Shannon diversity, but it is showing that there's greater alpha diversity of samples that fed on Didelphis compared to samples that fed on the other two. So kind of I guess like a last thing that I will mention. So we're seeing, so it's likely that at least some of the differences are being driven by host blood meal species. But also, we're kind of thinking about, okay, what else perhaps may be going on? And so something that we still need to delve a little bit more into, but so our colleagues in Panama, they actually found that there's a bit of a difference between the samples at Veraguas versus the samples at the canal site. So it could just be that is the reason that's driving some of these differences. So let's see how well you can see it on the screen. So the bug on your left-hand side, B. So these are both our pearlescens, but the ones labeled B were collected around the Panama Canal, and these are the most common type you'll see. versus the darker morph that's labeled C there. So on average, they tend to be a little bit bigger. They are darker. I had a colleague when we were in Panama collecting, and they were convinced it was like a completely different species. They're like, oh no, I've worked on these bugs for decades. I've never seen one this dark. So they looked at it a little bit further. So it is still our arborescent, but it's possible it may just be a subspecies. And so thinking about gut microbial communities and maybe some differences between the sampling sites at least could maybe be in part driven by some like host genetics. So something that we want to look a little bit more into. So just some of the overview of these results. We do see regional differences. So samples collected at Veraguas were significantly different than the samples collected at the 2 canal sites. And the canal sites have a more similar community composition than samples collected from Veraguas. And then also our 4 samples showed greater aquadiversity than our other type of habitats. And then our pasture and paradomestic palms have a more similar community composition than our 4 samples. And then finally, our samples that fed on Didelphis, the large American opossum, had greater alpha diversity and a different composition of bacteria compared to samples that fed on other mammal species. And then our Veraguas samples fed on a larger number of Didelphis. So ultimately, we do see some differences across region and habitat type. And we are seeing greater diversity at forested sites within Veraguas. And the differences at Veraguas may be driven by host blood meal species, vector taxonomy, and also perhaps landscape features. So I didn't touch on this really at all, but something to just look back on our map. So this circle is around the Panama Canal, but then where Veraguas is circled here. So it is quite a bit different. So something that we do want to look a little bit more at are what's happening at the landscape level, what's happening with different palm features. We did these like really big blocks of pasture, paradomestic, but something we can look a little bit more at is canopy cover, looking at organic matter and really kind of looking a little bit more at how the habitats look and perhaps trying to understand a little bit more what's driving some of the differences we are seeing. So moving on to my second half. So my next objective was to investigate the relationships between single and co-infections of trypanosome parasites and gut bacteria of arborescens. So I want to understand, are there any are single or co-infections associated with gut microbial diversity? So we've already talked about this first parasite, which causes Chagas disease, Trypanosoma cruzi. So it is acquired via infected blood meal. It replicates in the midgut. It transmits via an infected feces. There are some evidence that it does negatively impact tryptamine survival. So if they're only infected with Trypanosoma cruzi, some of their survival drops. And then also it is pathogenic to mammals. And then there's the second parasite, which is quite a bit less studied because it does not cause disease to humans. But so Trypanosoma rangelli is acquired via infected blood meal as well. It replicates in the gut and the salivary glands, and it transmits via the salivary glands. So more similar to a mosquito. There is quite a bit of evidence that it does negatively impact tryptamines, and then it is non-pathogenic to mammals. So arborescens are often found co-infected in Panama. And then, so I think this is like really interesting, but so co-infected tryptamines based off of a couple experimental lab work found that co-infected tryptamines have higher survival rates than tryptamines that are infected with either T. cruzi or only T. rangelli. So there's something about co-infection in the bugs. So taking our same samples that we had looked at before, they were also screened for T. cruzi and T. rangelli. And so ultimately, we found that 32% of our samples were only infected with T. cruzi. 7 of our samples were infected with only T. rangelli. 11.7% of our samples were co-infected with both parasites. And then about 56% of our samples were not infected with either parasite. And Because we had so few that were only infected with Rangella, we just didn't include them in the rest of our analyses. And then another thing that we looked a bit at is the age stages for the tritamines. We looked at some early stage and late stage. So you'll see a lot of figures that have results for both stages. There's quite a bit of like research as well as my own data that does show differences in early stage versus late stage tritamines. So in my samples, at least, our early stage tritamines had greater gut bacterial diversity than our later stage ones. So we're kind of thinking about them separately to not confound that information. So what we found, we're just looking at the bacterial diversity and either single or co-infections in the guts. So we found that our single infected and our co-infected samples displayed similar alpha diversity. So, so this single and co-infected samples are quite similar. And then so this right here, so this is thinking about beta diversity again, just it's displayed in a different way. So this is showing how similar or dissimilar the overall communities are to each other. So here we're again looking at our single and co-infected samples have similar community composition. And then, however, when you add abundance weighted, so this figure right here, we're not thinking about the abundance of the bacteria, but here we are adding abundance. So we're thinking about dominant versus rare taxa. And here we are seeing that single and co-infected samples are actually dissimilar when you add some abundance weighted to them. So there's something, so we can kind of think about driving to dominant taxa, driving some of the differences between our single and co-infected samples. And then now this is just looking at infected and uninfected. So we combine our single and co-infections and just comparing them to our uninfected samples. So here we're saying infected and uninfected samples have different community composition. And then when you add the abundance weighted to it, so we're thinking about the amount of bacteria of each individual taxa. So here we're seeing a difference in our infected and uninfected samples again. So, so overall, so we found that T. cruzi single infected and co-infected samples had similar alpha diversity, but different community composition and structure when we're thinking about our beta diversity. So here we can think that infection type may differentially shift the communities, but within sample diversity remains the same or is restored. And then also something that is, I need to kind of think about it a little bit more just as I continue to kind of put some final periods on my dissertation. But T. Rangella and T. cruzi do have different life cycles within the bug. So one thing that's really important is that T. Rangella does initially go into the gut and it starts to replicate, but then it moves out of the gut and goes to the salivary gland. So thinking about kind of timing and duration of an infection, that T. Rangella is in the gut less long than T. cruzi. So also, so that could be having some impacts on our single and co-infected. So perhaps I'm really kind of only sampling single infected, even when I think I'm looking at some co-infections. And something that I want to do next is look at burden. So how many parasites are we looking at for both parasites? And then also, so we're thinking about our infected, so that's either single or co-infected samples have different community composition than our uninfected samples. So a lot of this is driven by dominant and rare taxa. So this alone is just important thinking about as we sort of move on to vector control, like applied research, thinking about which taxa to look more at, perhaps which taxa to maybe target. In terms of, and also like just looking at the function too. So we have all the dominant taxa. What is it doing in the bug? With my results, I can't tell you what it's doing. So something to look at. So some of the dominant taxa as well as the rare taxa. And then so finally, so overall, so tritamine gut diversity differs across region and habitat type. And we see that infection can alter gut communities. So I'm also finishing a lot earlier than I thought. I always talk a little bit quick when I'm up here. But so I think I just kind of want to stay here just for another second. So stepping all the way back. So again, what I was really most interested in, are there any associations between disturbance and the gut microbial communities? And then if so, what is that like? Are there any kind of impacts further down on their ability to pick up infection, maintain and transmit infection? So kind of the next step with a lot of this research, I really want to start looking into function of the gut microbes. So we're seeing some differences in the communities, but ultimately it's really hard to say what that means. Is it important? Maybe, maybe not. Thinking about less diversity, how much like less diversity do you need to have any kind of maybe downstream impacts on the insects or the ability again to acquire the parasite? And then also to just functional diversity. So perhaps there's a lot of functional redundancy happening, and then maybe some reduced diversity doesn't have very many impacts on the insects themselves. So something that is a long, it's something I'll have to do like quite a bit later on, but something that I'm really, really interested in. We're seeing these differences. Exactly what does it mean? Can we track anything? And then ultimately, think a little bit more about vector control management and any kind of intervention ideas we can come up with. So moving on. So I really did think I was only gonna have like 2 minutes left. So I have the next section is very, very brief. So I'm happy to answer as many questions as y'all have, and I can kind of go into a little bit more detail, but so I'm happy to be starting my postdoc here. And something that I really want to kind of continue for my dissertation research in here is really thinking about disturbance and what impacts it has on microbial communities, and ultimately what that may mean for infectious disease transmission. And also, I really want to try to think about my research more in a One Health framework. So One Health framework is really combining and thinking about the importance of health between humans, animals, and the environment. And too often the One Health framework leaves out plant health. So this is an expanded One Health framework where it's including plant health, which is also just as important as animal, human, and environmental health. And then, so I'm hoping that for my research, while I'm here, I can think about disturbance and plant health, namely kind of thinking about the microbial communities of plants, and then what that means for infection. So largely, I would like to investigate the effects of urbanization on plant microbiome structure. And when I say microbiome structure, I really want to think about, or I guess explore, bacterial and fungal communities, and then also looking at some functional diversity as well. So that's something that will be new for me, but I'm excited to kind of learn some more sequencing techniques and bioinformatics, and also work with fungi, which I've done very little with. And then also how pathogen burden affects plant microbiome structure. So a little bit like I talked about in my dissertation, but really thinking about both disturbance in the form of urbanization on microbiomes, and then also infection on the microbiomes. So I will, I'm excited to be working with the Fenjikowski lab. And so we'll take like my 2 questions that I need to expand quite a bit more on. I'm really just getting started here. But so in the lab, they study plantago, which is a widely distributed perennial herb commonly found in human disturbed landscapes. And then they also work with this plant pathogen, which infects the plantago, a powdery mildew fungal pathogen. So thinking about my kind of 2 questions is both urbanization and looking at urbanization gradients and what the microbial communities look like, and then perhaps doing some experiments looking at different like levels of urbanization, and then also looking at infection burden with the plants. So looking at different amounts of burden, and then how that perhaps shifts has an impact on the microbial communities. Let's see, and then also, so I'm hoping quite a big chunk of my postdoc will also be dedicated to STEM K through 12 community outreach. And so I do have more time, but so I could talk a very, very long time, and I would love to chat with you afterwards. But just so STEM K through 12 and community outreach is really important. And something that I really want to do with my advisors is really thinking about how we can best link there's so many universities in St. Louis. I didn't realize how many there were, but really trying to link a lot of the knowledge, a lot of the resources with a lot of the schools, particularly that have like marginalized students, and then even more so schools that are under-resourced. So how can all this university, again, like knowledge and just resources, really kind of make that connection, and thinking about how many students just don't have access to just environmental sciences, thinking about environmental careers. And then also, and I really want to try to expand it out to the community and not just work with schools, but hopefully their parents, their family members, just other community members, and really kind of thinking about like science literacy. And there's so much research that's happening just like in this area alone. And a lot of it has direct impacts on the folks that live here. And I don't know how many people know what research goes on. So I really want to try to like work to make these links to just to give people the ability to advocate for themselves, so they know what's happening in the communities, what's happening in their own neighborhoods. And then they can then truly just like know and you know what the government's doing to their own neighborhoods, how to like maybe vote differently, or how to kind of like move differently if there's just sort of like more knowledge transfer. And then I think also too, especially for the university to have the knowledge of the local communities. There's people who've been here for generations, and but I know there's so many transplants to universities, myself included. I'm from California. So also learning from the folks who have been here, and also knowing what's important to them, just learning about the local and cultural knowledge of St. Louis and the surrounding areas. So with that, so I'm really excited to work with all three of my advisors. Rachel, who is with WashU, and then Solmy, who's with Tyson, and then also Daniel Lee, who's with Southern Illinois, Edwardsville. And then also working with the St. Louis Urban Ecology and Evolution Working Group, and also with Tyson Research Center. There's already a lot of really cool outreach happening. So I'm really hoping I can kind of like learn from everyone, and then sort of see where I can fit in, or how I can sort of add to all of the really cool events happening here. And that is my talk. So I went a little bit longer, but I really appreciate y'all, and thank you so much for listening. And I'm happy to answer as many questions either here or afterwards. Thank you.
Great, thanks so much, Kaylee. It's really exciting to see the microbiome research happening at WashU, and folks taking it from a lot of different angles. So we have plenty of time for questions. I'll come around with the microphone if anyone in the audience has questions.
Thanks so much for the wonderful talk, and welcome to campus and the department. I actually have a very sort of basic question on microbiome sequencing and biology. Why do you use sort of 12S like the short subunit versus 16S the large subunit for those different modes of sequencing? Like, what's the inspiration behind that?
So I don't have a great answer for you. I did a lot of the work with one of my older lab mates, but the 12S can target targeting the vertebrate DNA sequences, so there we can kind of figure out what the host species are, versus the 16S is targeting the bacterial DNA. So that is the best answer I can give you. I can't go into like any more detail, but yeah, thank you.
Thanks for the talk. This is more of a natural history question. Why do the bugs, so you said you can find the bugs in the royal palms, Do they mostly live in the royal palms? Or, and so what, is there some type of special association with those plants? Just very curious about that.
That is a good question. I don't think I know quite about the association, but so the adults have wings and they can move around, but they tend to just stay in the palms. So I suppose there's just enough resources. But if they need to travel, they can crawl, and we've done some like flight experiments and seeing how far they can fly, but they tend to just stay in their palms.
And then prey on the mammals that.
Yeah, and the mammal, yeah. So sorry, yeah. So yeah, so the mammals get to the palm crowns, and that's where they feed from them.
Perhaps some questions from the YouTube audience.
Thanks, Kaylee. Indeed, we do have questions here from YouTube. Kim Medley is wondering, could the darker kissing bugs get warmer in sunlight or adjust their behavior to avoid sunlight that impacts their blood meals and microbiota? Or could warmer bodies in darker morphs or subspecies affect the survival of certain gut microbiota?
That is something that I had not considered. I think that's a good question. I would say perhaps, yeah, and I think that's something that we can definitely look a little bit more at. I saw that there are different morphs, and then that's kind of where I stopped. So I appreciate your question and kind of thinking about maybe what that means for the gut bacteria and the host species. Yeah, thank you.
Kaylee, great talk. Yeah, it's a very important topic. I just had a question out of curiosity. You mentioned something about like the anosome affects the survival of the triatomine. Do you know what is the prevalence of the parasite on the bugs? Is it like is there like this is a different question, like coevolution among like parasites?
Them like co-evolving with the parasite and the bugs.
Yeah.
Yeah, I don't know too much about like the evolution part of it. But I can't touch if this was a part of your question. But so looking at some of the lowered survival, so the parasites likely compete against some of the like symbiotic bacteria in the gut. So that could be having some impacts on survival there. But in terms of, yeah, I can't fully answer your kind of evolution question. But they have been infected and co-evolved for a long time, but in like anything more than that, I'm not sure.
Yeah, thanks for a great talk. In the first part, it looked like the beta diversity among the kissing bugs within the Baragua site was larger than among bugs at the two canal sites. I'm curious if you have any thoughts on what's driving that pattern. And it seems especially interesting in light of the blood meal analyses, where if I understood correctly at the Baragua site, there were fewer hosts that the kissing bugs were feeding on.
Mm-hmm. So for your first question, yeah, so that's something that I haven't like looked a ton into, but kind of like I guess a next step that I need to look at is let's see, some like spatial information with the palms. So as I had mentioned, so they spend like quite a bit of their lives in the palms, and we are seeing, I didn't talk about this at all, but bugs that are from the same palm tend to be very similar, so it's possible, kind of thinking about how many bugs from each palm in my sampling, and then perhaps maybe that's why there's some more distribution in Baraguas. I might have been sampled more palms there. So that's something that I need to look at to kind of get at more of those differences. And then you said also thinking about the blood meal, like why there's the differences, or
Yeah, I was just thinking maybe you might expect more within site beta diversity of the microbes if they're feeding on more different species of mammals.
Oh,
It
right.
seemed like there were fewer species of mammals
Yeah.
that were feeding on at the Baragua site.
yeah, I'm not sure. Yeah, no, I really need to like dig more into all the kind of blood meal stuff, and then again like palms and kind of like which palms they're actually from, and then also too there's like clusters of palms, and kind of really rethinking how I'm even like defining like the sampling sites and et cetera. Yeah, thanks.
We have a couple more questions here from YouTube. First, Kim says, thanks, Kaylee. And she has an additional question related to your upcoming Plantago work. Are there any insects that utilize Plantago for oviposition whose microbiota could be impacted somehow by Plantago fungal infection?
That is a good question. Yes, perhaps, yes, yeah, I'm looking at Rachel, I was like, I have a very surface level knowledge at this point, but yeah, there's definitely something to, yes.
Great, and the second question is from Cheyenne Anderson, but it's a version of a question that was just asked. I can ask it again, see if you mind. If I understood correctly, I was surprised that forests had less host diversity. I would assume forests have greater diversity of hosts. Is there any hypothesis?
So I'm not sure. Something that I also didn't touch at, and I don't have the data kind of like off the top of my head, but so for the blood meal species, we also only looked at the dominant sources. So I was really just kind of flattening out to like one host species, but they can feed on, or we were able to identify up to like 5 different genus of the vertebrates. So it's also possible there is actually more richness, but then it kind of gets flattened out to looking like less. So I am trying to figure out how to really kind of analyze the data in a way to look at some more of that diversity of the host. So it's possible that's not even true. So I think that's probably my best answer for you. Because we just being in the field, we would see so many vertebrates. So I would definitely expect it to have more. So kind of looking a little bit more at the rest of our data to see how true that is, or if I really just kind of flagged out all of important information. But yeah, thanks.
Thank you.
All right, thank you. I have a question, and I promise it's not about Plantago. My question is, so these bugs are spending a lot of time up in the palms. Obviously, they're also important from a human health perspective. And so I'm wondering, I know this was not a focus of your research, but what are the rates of Chagas disease in these places where you're sampling? Is that data that's available from any like health departments? And then also just, basic question for the next time I teach disease ecology. So you can help me understand. So they're in palm trees, and then like, are they seeking out a better habitat somewhere else when they come down and then end up biting a human, or like what causes them to leave a palm and become a problem from a human health perspective?
Yeah, so I guess for your second question, likely, yeah, I guess if they're searching for a host, and then they'll leave the palm. And then if they like happen upon a human, also there's a lot of palms being built near homes. So also like, so them finding another palm, and then it's like really close to humans. So that definitely is like an issue. And then sorry, your first question was, oh no, I forgot about. Oh. Oh, yes. So that information does exist. I apologize. Sometimes I don't have that information. It's not, no, I would make something up. Yeah, it definitely is a problem. And there's a lot of differences between looking let's see, where in Panama. So that's something that my old advisor and a lot of the current grad students are looking at to see how much infection there is depending on kind of where you are across Panama. But yeah, I don't have that answer.
So if I'm remembering right, there's unequal infection with T. Rangeli. So is it possible that if I get bit and I get infected with T. Rangeli, which isn't pathogenic, that could give me resistance to the pathogenic species? And I'm wondering then, I'm personally thinking meat, but I'm also thinking in terms of the marsupials and rodents that are getting bitten, could that explain the lack of coexistence in the hosts?
Yeah, I think so. I don't know too much about kind of the human side of it all, but I would imagine they would be interacting and maybe competing still. And perhaps that would yeah, maybe have some type of protection a little bit, but I'm not really, I'm not sure. That's really interesting to think about.
I have a kind of a related question about the co-infection. So also great talk, thank you. So I'm forgetting the not cruisy species, the other species.
Rangelli.
Rangelli, thank you. So you said it clears the gut and ends up in the salivary glands, right? So how confident do you feel in like your samples came from the gut?
Yeah.
So you could have some uninfected samples or that like maybe had this infection before and it's cleared and gone to the salivary glands. So like if you went back to do this, would you like adjust, if you were interested in the co-infection, would you adjust like how you're collecting the samples or testing for that infection?
Yeah, there's a few things I would do. So we are detecting the parasite in the gut, but like I don't have like burden, I don't know how much or like active wise. So it's really hard to say. It's like it was there at some point. I think so. I do have a bunch of salivary gland. So I dissect salivary glands with the help of a lot of undergrads and also sequence them. So something that so one, I would look at burden within the gut, kind of if I'm really thinking about the co-infection within the gut, but also two, looking at the salivary glands and saying like it was there, and I don't know. I think at least like to just give some more information about the presence of the Rangelli and the bugs, but really the biggest thing I just need to do some like quantitative PCR, which I thought about doing in December. I was like, I could either go home or do lab work for another couple weeks. So, but it definitely is on my list to kind of think about. But thank you.
This is an evolution adjacent question. I hope that's okay. So do people know how closely related the two trypanosoman species are? I'm also wondering how much vector specificity is there for each species. Kind of figure out like why are there two if they're both specialized for this one insect possibly?
Yeah. So they are very similar. The two parasites. I don't, I'm actually, I don't know. No, it's fine. Yeah, no, it is something that it is, no, I think it is a good question. Something that it's good to think about. But no, I'm not sure, but they are quite similar. And then there are, I know at least in I think a lot of like human work where it's easy to detect both pairs, like you're trying to look for one and you end up detecting both of them. So they are quite similar and you need to have really like specific like tests for them. But in terms of like, yeah, I not kind of evolution side, but yeah, thank you.
We have a question from Suzanne Renner. Chagas is transmitted by other bugs as well, such as Triatoma infestans. Did you look at any other bugs too?
So in Panama, there's a second species that does carry the parasite, another kissing bug. So I had a whole chapter looking at Triatoma dominiata. And so I just didn't touch on it today as I thought I would run out of time. Yeah, I mean, I guess the biggest thing I can say is that we are seeing differences in the gut bacteria between the different species. We didn't collect a ton of the dominiata, so I don't have like a lot of results with it, but yes, we did. We have two species, and that's kind of an ongoing project.
Do we have any more questions? All right, well, we can always continue conversation next door at the reception, where we'll have some refreshments and we can chat with Kaylee about her past and upcoming research. So let's thank Kaylee once again.
Thank you.